everyone, and welcome back to week number 3 of our Molecular Biology for Engineers course. This week we're going to be talking about the structure and function of RNA and proteins, and kind of following up a little bit with some of the stuff we talked about last week, and then getting ourselves ready to go right into transcription and translation next week as we move forward. So today let's get started with looking at the structure and function of RNA. Now we know that it's very similar to DNA, and we really explored the structure and function of that one last time. So let's look at some of these differences with RNA, and we'll talk a little bit more about the key roles that it plays in these very important processes that are happening within the cells. So first of all, we know that it is a pentose sugar, just like we saw with DNA, but unlike DNA, it's got a hydroxyl group right here on that second carbon of the pentose sugar, instead of the oxygen that we find in deoxyribose. So we've got our ribose sugar, we've got a little hydroxyl group there on that second prime carbon. So now we're going to see uracil coming in and being substituted for thymine. So instead of a T, we're going to have a U. It's going to bind with our A most of the time, but sometimes we are going to see it binding with guanine instead. So every once in a while you might get a GU interaction. Unlike DNA, this is a single-stranded molecule, and that's pretty important as we saw with DNA replication last week. It'll also be important for these processes of transcription and translation that we'll be looking at more next week. So looking at some of those different processes that RNA is involved in, let's start with transcription. We're going to go into a lot of detail about how this works next week, so I'm not going to do too much with it right now, but what you should know is that the DNA strands serve as a template for this process of transcription where we start to build the single-stranded RNA molecule. And the RNA is going to be formed in the 5' to 3' direction, just like we see in DNA synthesis. When we look at translation, we know RNA is going to be involved as sort of directing the whole process of protein synthesis. And again, we're going to talk about the details of that a little bit more next week. And then of course DNA replication we went over last week. So just to kind of remind you, we know that RNA polymerase plays a major role in this whole process of being able to replicate DNA. So if you don't remember that, you might go back and review it. Another set of important things to remember about RNA is that they come in a couple of different flavors. And we're just going to kind of step through these really quickly. So you've got messenger RNA, which is going to play a pretty key role in some of these processes that we are going to be talking about next week. And this is abbreviated as mRNA. So it's going to be something that's involved in carrying messages to be able to direct some of these different processes. You've also got transfer RNA, or tRNA. This is going to be something involved in the transfer of information. We've got ribosomal RNA, and this is going to be found within the ribosomes of the cell. We've got small nuclear DNA, and just like the name implies, it's found in the nucleus. But it also plays a major role in RNA splicing by helping to make up some of the spliceosome machinery. And we're going to be talking about that a little bit later in the semester. So just know for now that it's involved in RNA splicing. We've also got the microRNAs, or the miRNAs, and these are going to be involved in protein synthesis. So again, you're going to be seeing these come up again next week. We've got the small interfering, or the silencing types of RNA. Now these are also known as siRNAs. And they're pretty unique in terms of the way that they look and the way that they behave. Number one, they're double-stranded. What the heck is that about, RNA? Small interfering or silencing siRNAs. And this one's going to be involved in kind of messing up some of the processes that we're going to be talking about next week. So what it does is it comes in and it degrades some of the material within our old friend, our mRNAs, or our messenger RNAs. And by doing that, it disrupts transcription, thereby preventing translation and preventing the synthesis of proteins. Therefore, it is involved in gene silencing and prevents the whole process of protein synthesis whenever it's involved. And we'll talk a little bit more about these siRNAs later in the semester as we start to get into some of the applications for different biotechnologies. So, with that, maybe we're ready to go on and start talking about proteins and look at their structure and function. All right. All right, so let's talk a little bit more about proteins. We know that they make up about 50% of the average cell, and this is just sort of in a typical situation. But you've got tens of thousands of proteins hanging out in your body right this second, like right now, just hanging out. doing all kinds of cool stuff. So they have a lot of different functions in the human body and in the bodies of other organisms as well. So we can see that they play roles in things like growth and tissue repair. They're really important for breakdown in terms of breaking down metabolic products and breaking down things like food in your digestive tract. They also help to speed up reactions. So one of the major roles of proteins in the body is to function as enzymes. And we're going to talk just a little bit more about enzymes here in a little bit. They also have a major role to play in immune responses because all of your antibodies, those are proteins. So all of that you know about innate immunity versus acquired immunity, we're talking about antibodies that are made of proteins. They also play a pretty major role in transportation of materials. One really good example of this is that your red blood cells have a protein called hemoglobin, and hemoglobin is really important for the transport of oxygen and carbon dioxide and the exchange of gas for cellular respiration, and also for getting them to the lungs for actually expiring and inspiring. They also play a role in communication in the body by helping to make up hormones and do things like protein endorphins that are really important for making you feel good after you've been working really hard. And they can play a role in regulation as well. So anything that's coming in or out of the cell that's not just diffusing through the phospholipid bilayer is probably coming in through some sort of regulatory protein channel. And those are going to be very specific things that we can talk a little bit about later in the semester. But for now, just know that they serve as these little doorways or these little gateways to sort of regulate what comes in and out of the cell. Now, proteins are made up of what we call amino acids. And we're going to talk a little bit more about some of the different kinds of amino acids here in a moment. You probably have already heard of this and are a little bit familiar with it, but we'll go over it just a bit anyway. So all of our amino acids are going to be made up of three major components. You've got an amino group, you've got a carboxyl group, so this is your amino, this is your acid, that's where we get our names for this. And then all amino acids have what's called an R group or a radical group. And this is radically different with each of the 20 different amino acids. And that's what characterizes an amino acid as being the specific amino acid that it is. So we're looking at that R group to figure out which amino acid we're dealing with. And with amino acids in the human body, we can't produce all of the amino acids that we need from things on our own. So there are some amino acids that we're going to have to take in through our diet to be able to build all of these different kinds of proteins that we need. And the amino acids that we have to take in through our diet, we call the essential amino acids. And there are 9 of those. We'll look at those here in just a moment. But first, let's talk a little bit more about the structure of proteins. So we know they have this three-dimensional shape, and we'll talk a little bit more about shape here in just a second. They also have this ability to be reversible, so they can change their shape to be able to perform a function and then sort of snap back into the shape that they were in originally. And this is especially important for enzymes, but also for other types of proteins in the body as well. There's also this process of denaturation, and this is bad. This is when we alter a protein to the point where it doesn't have that ability to snap back anymore. So you can denature a protein by doing a couple of different things. You can expose it to extreme heat, and then the protein will unfold and no longer be working. You can think about like an egg. Whenever you go to cook an egg, that white part of the egg is all albumin, so that's a type of protein. And what happens when you heat the white of an egg? It drastically changes, right? And once you've cooked an egg, you can never get that albumin to go back into the snotty, clear white stuff that it was before you cooked it and made it the solid white deliciousness that it is. So denaturing a protein by heat is something that's irreversible. You can also denature things by adding an acid to it and changing the pH to the point where it destroys the structure of the protein. An example of this that we use to our advantage all the time, or at least if you don't, you probably should, is when we're trying to prevent browning. So there's an enzymatic reaction that happens when food's brown. Let's take, for example, an avocado. We know when we slice an avocado open, it gets exposed to the air, and without too much time, it starts going from this beautiful, luscious, delicious green to this kind of dingy, brownish, green goop stuff that you're not sure you want to eat, right? So what do we do to try to prevent that from happening? Well, you can squeeze a little bit of lemon juice or lime juice on the surface of the avocado, and that will slow the browning process. And the reason is because it's destroying some of those enzymes that are going through that enzymatic reaction that causes the browning process. So by adding lemon juice or by adding lime juice to your guacamole, you're slowing down that enzymatic process by destroying some of the proteins that cause that reaction to happen. You can also have other things that cause denaturation, but heat and pH are kind of the two biggies. You can get it from other extremes or from adding or taking away different things, but we're not really going to go into that too much right now. Now, to create proteins, we want to take those little amino acids and we want to stitch them all together into these big polymers that we've been talking about. And we call these polymers peptides. And that's because taking one amino acid and another, we bind them together, and that bond is called a peptide bond. And this peptide bond is going to be something that occurs between that radical group and the carboxyl group. And these peptides are going to be named for the number of amino acids that are involved in that whole process. So if you just have 1, 2 amino acids together in this little peptide bond, now we've created a dipeptide. If you've got 3, a tripeptide. As you start adding more and more, you're going to get what are called oligopeptides. And eventually, when you have a lot, we'll call it a polypeptide. And when you have 50 or more of these amino acids strung together, that's going to be what we call a protein. Okay, so let's continue talking about the structure of proteins. We already said that they're made up of peptides, but what happens after we start stringing things together is that we've got to get them into a small space, kind of like how our DNA needed to be compacted to be able to fit in the little places that we keep it in. So we're going to start folding these proteins to be able to make them more compact. Our first structure, or what we call a primary structure of a protein, is just going to be a sequence of those amino acids kind of strung together. And so we're going to have these little polypeptide sequences with, again, 50 or more amino acids making up some of our most primary structures of proteins. The secondary structure is when we're going to start to coil and fold these proteins together. And this is held together by hydrogen bonding. And we can see it occurring in a couple of different formats. You have what's called an alpha helix, which looks very similar to our DNA, so it's got a spring-like or kind of a coiled appearance to it. And the other type is what we call a pleated sheet or a beta helix type of formation, where the amino acids are kind of put together in more of a folding sheet type pattern, as opposed to a spring-like pattern that we see with the alpha helix. Tertiary structure is where we're going to continue to fold that protein, and this is going to be held together instead of by hydrogen bonding by the interactions of hydrophobic and hydrophilic molecules. And then we're also going to see some van der Waals forces coming into play here. And I'm not going to go into a lot of detail talking about van der Waals forces. They're these like little weak associations that are happening between little pieces of the molecule. But I do have a supplementary video that I'm going to send to you guys along with this video so that you can go through and learn a little bit more about van der Waals interactions. In this tertiary structure, they come in again 2 different types. We can see what are called globular proteins, and this is just more round kind of shaped proteins. Or we can make them more fibrous where they're a little bit more strung out, kind of like a fiber and elongated. So we can see those 2 different kinds of things happening. Now, not all proteins, but some are going to move into sort of a 4th stage or a quaternary stage, and this is a type of structure that's going to involve 2 or more of these polypeptides together. And these are going to be held together again by these hydrophobic, hydrophilic forces, but also by ionic bonding. And a good example of something like this is our hemoglobin molecule. Okay, so let's go back for a second when we were talking about essential amino acids earlier. So when we say essential amino acids, again, these are amino acids that we need to acquire through our diet. Sometimes in the literature, you'll see these referred to as indisposable types of amino acids, and that's because we really need them to be able to function. So there are 9 essential amino acids that we have to get in through our diet, and we've got them listed out here. I'm going to expect you to be able to list these out. I'm not going to expect you to be able to name all 20 of them or to draw their structures or to know what their distinct R groups look like or any of that kind of thing. And if you had to do that for biochemistry, like, I'm proud of you, but I'm not going to require that for this class. But I do want you to be able to recognize which ones are essential versus non-essential and some of these other things that we're going to talk about here in a second. So our essential ones, we've got histidine, isoleucine, leucine, lysine, methionine, threonine, phenylalanine, tryptophan, and valine. So those are our essential amino acids. You can also have what are called conditional amino acids, and normally these are non-essential types of amino acids, but when you're sick, you're not really getting enough of these a lot of times, especially if you're limiting or restricting your diet. So these are the types of amino acids that can become kind of necessary to try to take in more through your diet when you're not feeling well. So those are our friends arginine, cysteine, glutamine, glycine, ornithine, proline, serine, and tyrosine. And when we're talking about foods and getting some of these amino acids, you might see the word complete proteins being thrown around. And when we're talking about that in a nutritional biological sense, in a research sense, a complete protein is going to be a type of protein that includes all of the things that we need in terms of amino acids to be able to continue to synthesize the proteins that we have inside of the body. When we're referring to those complete proteins, we're talking about something that we're going to eat that's going to contain all of these essential amino acids that we need to be able to properly function. And when you're looking at complete proteins, you'll see references to things like net protein utilization, which is how much of the protein we're able to actually use biologically in the body. And then also you'll see the term limiting amino acid. And this is the lowest amount of an amino acid that's found within the complete protein. So it's still present, it's still part of a complete protein because it has this amino acid, but it has maybe less of this particular amino acid. So for example, when you look at things like wheat and corn, those are going to be complete proteins, but lysine is going to be the limiting amino acid because it's found in the lowest quantity within those kinds of food groups. However, if you look at something like legumes or some of these different beans that you have, they're going to have a different limiting amino acid. So in some places in the literature, they talk about combining things like rice and beans because then you're changing up the limiting amino acids and be able to get some good net protein utilization out of eating those complete proteins. So for those of you who are from cultures that traditionally have beans and rice, or for those of you who just enjoy food that comes from cultures that have traditionally included beans and rice as part of the cuisine, you're getting your complete proteins and you're making sure that you're taking care of that limiting amino acid problem when you do so. So go out, eat some beans and rice, because it's delicious. And on that note, that's better. Okay, so we're going to wrap this up by talking about enzymes. So an enzyme is basically a biological catalyst, and again, these are proteins that make these guys up. So what they do is they help to lower the activation energy of a variety of different reactions so that that reaction can take place. It helps to really speed up the process and get things going. And proteins are named for whatever it is that they're acting on, and that thing that they're acting on is what we call a substrate. The name then has the suffix ase or ACE on the end to indicate that it's an enzyme. So for example, if you come across a protease, that means that it's an enzyme that breaks down proteins. A lipase is going to be an enzyme that breaks down lipids. As we've seen, you have things like DNA polymerase and RNA polymerase, and those are all enzymes as well. All enzymes are going to have an active site or something called the binding site. That's where you're going to see some of these things interacting with the substrate, and also with different types of regulatory molecules. So within the body, these enzymes can really get to working fast and they can make things happen. So they have to be really tightly regulated to make sure that they're not running amok and messing things up. So that can be done in a couple of different ways. First, you've got regulatory molecules like activators and inhibitors that tell these enzymes when to start and when to stop. And you can have these little regulatory molecules that are going to bind onto the enzyme to be able to either activate or inhibit it. You can have some things that bind directly to the active site, which prevents the normal substrate that the enzyme would interact with from being able to bind and thus preventing the reaction from happening. That's called a competitive inhibitor. You can also have an inhibitory molecule that attaches to someplace other than the active site of the enzyme and somehow changes the shape of the enzyme so that the active site is no longer available for the substrate to be able to bind. And that's an example of a non-competitive type of inhibitor because it's not directly binding to the active site. We also see compartmentalization playing a role in the way that we regulate enzymes. And this is pretty important. Some enzymes need to be kept at a certain pH to be able to function optimally. or they need to be able to be kept at a certain pH to keep them from functioning until we're ready for them to function. So they can be sequestered in various organelles within the cells of the body to be able to try to sort of sequester them away, compartmentalize them in a place where they can be stored safely until they're ready to be used. And another thing that is involved in the process of enzymes are what we call cofactors. And these cofactors can play a major role in helping the enzymes to perform their functions. In fact, some of these enzymes won't work at all unless you have a cofactor. For example, DNA polymerase that we talked about last week requires magnesium ions to come in and act as cofactors so that it can go through that whole process of DNA replication. And in fact, 2/3 of all human enzymes require some sort of cofactor to be able to perform their duties. And cofactors can be a lot of different things. Sometimes there's a special kind of cofactor that we call a coenzyme. And these are organic molecules that are derived from water-soluble types of vitamins. A good example of this coenzyme relationship is through the building of collagen, which is a protein that's really important for lots of different things in your body. It helps to make your bones strong, it makes your whites of your eyes white, it makes up a lot of the different types of tissues that are found throughout the body, but it can't be synthesized without vitamin C. So in this case, vitamin C is serving as the coenzyme for the production of the collagen protein. And that sort of brings us to the end of this lecture. So maybe we should just do a little bit of house cleaning. All right, just a couple things to sort of wrap up this third week for you. As always, your highlights are going to be due on Friday by 5 p.m. Make sure you get them to me so I don't have to take off points for lateness. This week, your highlights are going to include stuff from the readings, which were pretty minimal. Really only talk about the stuff that has anything to do with RNA and proteins, which is like very little of that chapter. So don't go into like any of the other things that we're going to talk about next week from the chapter. Obviously, you're going to want to include some of the highlights from this lecture video, and you want to include some highlights from the videos that I sent you guys links to in the first email this week on Monday. So make sure that you watch those videos and generate some of the major takeaway points from that. This week, now normally with highlights, you need to limit it to 1 page because we're working on science communication and being concise and all of these good things. But this week, you're going to have an additional 2nd page in your assignment in your highlights assignment. And this is going to be an assignment that we're calling the amino acid assignment. So on that 2nd page of your highlights, just send it all to me in one document. You're going to choose an amino acid, any amino acid. Pick one, any one, take something that you're interested in. I want you to describe the structure, so make sure you talk about the R group because that's what sets it apart from other types of amino acids. I want you to list the proteins that this particular amino acid can be found in, and it's not like you have to list every protein out there. So just give me, like a couple of proteins that you can find this amino acid in. And then if you pick an essential amino acid, I want you to talk about the dietary sources of where you can get that amino acid. If you pick a non-essential amino acid, then talk about how it's made in the body. What is the process for synthesizing it, or the pathway or the mechanism that's involved there. And finally, I want you to talk about how this specific amino acid relates to human health. And that can be very broadly defined, so you don't have to spend a lot of time talking about it. Just think about what that means to you as you're doing your research, and talk about how it is related to human health in some way, whether it's in some sort of disease, whether it's in the way that we process something on a physiological level, whether it has something to do with molecular medicine. That's totally up to you. So sort of just broadly think about how your amino acid relates to human health in some way. Some other things that you should be thinking about: we are going to start having a series of guest lectures coming up after the first exam. So we'll be talking in the coming weeks about how that's going to work. With this being an asynchronous course, what we'll do is whatever scheduling works for the guest lecturer, that's when we're going to schedule them. And I would like for as many of you to be there for the live session with that guest lecturer as possible. But if it just absolutely won't work with your schedule, that's fine. We're going to record the sessions and you can watch them later. And those guest lectures will be things that you'll include in your weekly highlights when we get to that point. So So just be aware that that's going to be coming up after the first exam as we start getting into different biotechnologies. Another thing, I see that some of you have been doing a really good job of getting in on those discussion boards and starting some good conversations. I'd like to see more of that, and I'd like to see more of you participating. So please get into those discussion boards, make a comment on one of the prompts, or ask a question, or just try to get to know your classmates by engaging in some of these discussions of things that we've been talking about in lecture, or that you've been watching in videos, or that you've been reading about in different assignments that we've done. So get in there and start talking to each other. It'll be fun. And I'm not going to comment on anything in the discussions, but I will be lurking and watching and seeing who's there, and also kind of paying attention to what kind of conversations you guys are having. And that may be something that comes up like as a bonus question on an exam later on. So do be following along and do be keeping up with those discussion posts. So I hope that you all have a really great week, and I will see you next week when we start talking about transcription and translation. Bye!